Good afternoon, everyone. It's April 14th, and we're just a few days away from the end of the semester. And I'm very happy to welcome you to the second journey lecture and the last journey lecture of this spring winter semester. And I'm very thrilled to introduce our colleague, Professor Laura Scott. And as you know, the tradition has been that a mentee and a disciple of the journey lecturer introduces the journey lecturer. And so I just do the honors of introducing Laura and introducing the disciple. I don't know whether Laura knows who the disciple is because it's supposed to be a surprise. Laura came from a different background. Laura has a PhD in cell biology from Johns Hopkins in 1993 and then did an MPH here and started as a postdoc in 2000 in the statistical genetics group and then has been a faculty member since 2003. So I would like to welcome to the podium Sarah Hanks, our brilliant doctoral student working with Laura for many years. But I also want to tell you that today is a very many is quantified, like, not too many, just enough. And I just wanted to say that today is a very auspicious day according to the Bengali calendar, because today is the Bengali New Year. And so it's like, but we have a different lunar calendar than the other lunar year that was celebrated. That's why I'm in red. But I think with that auspicious moment, which brings us all together this afternoon and also brings a mentor and a mentee together, let us welcome Sarah.
So today it is my absolute pleasure to introduce my PhD advisor, Dr. Laura Scott. Since joining the department as a faculty member in 2003, Laura has made numerous significant contributions to the field of statistical genetics, including the identification of genetic variants that increase risk of common diseases and the development of methods for GWAS as well as the analysis of genomic and functional data. And she is currently a PI or co-PI of multiple grants seeking to understand the factors that influence gene expression and regulation in type 2 diabetes-related tissues. On top of her scientific work, Laura is a leader of diversity, equity, and inclusion efforts in academia, establishing the Biostatistics DEI Committee in 2017. As its chair, she organized a DEI research seminar series and led efforts to promote DEI learning and intercultural exchange. She is also involved in other university and community-based organizations with a focus on racial equity and social justice. On a personal note, I have been Laura's student for exactly the right amount of years. And under her mentorship, I and all of Laura's students have learned to turn statistical analyses into compelling stories about human biology, to persevere through difficult but worthwhile challenges, to listen openly and thoughtfully to others with different perspectives from our own, and to make all of our scatterplots exactly square. For me, what shines through in everything that Laura does is the deep and genuine curiosity about the world around her. She never stops asking questions about anything and everything, from what programs of gene regulation define cell types in the human body, to what university policies ensure that all individuals are valued equally for the work that we do. This curiosity, which is a hallmark of both a great scientist and a valuable community member, is a trait that I hope to inherit in this academic pedigree, and that I think we would all do well to emulate. Please join me in welcoming Dr. Scott.
Thank you very much, Ramar and Sarah. I think, Sarah, that was the nicest introduction I have ever gotten and probably ever will. Very neat. So I want to tell you today about my journey, and it's a journey that's different from most people in the room, in that I came here through biology, cell biology. And so at my heart, I'm a patterner. So I like patterns in trees, bricks, light cells, data, people, anything that I see. I also have an internal sabbatical clock. So I just get bored after like 7 to 10 years of doing the same thing. And at this moment, I happen to be a statistical self-analysis. So I'm going to tell you about some different partitions of life. So from early years, cell biology, epidemiology, different phases of statistical, genetics, genomics, cell biology, welcoming my son who is here today, Theo Scott, and a little bit about DEL. So this is part of the journey, and you can see that it's kind of interesting. So I really like this quote up at the top. So it says, I don't know how to speak Latin, but omnis cellula a cellula, which means everything begins and ends with the cell. And so you can see my journey here, starting with chemistry, cell biology, epidemiology, statistical genetics, making its way back around to statistical cell biology. Now, if you had asked me when I started, if this was the journey I was going to take, I would have had no idea. So this journey to make it has taken me many different places. And I just wanted to give you a sense of what going on a journey like this looks. So the yellow dot is going to indicate where I was. I started at home. The black circle indicates both Clinton, Michigan, where I grew up, which is 22 miles from here, and Ann Arbor. So this is what happened in my life before I came back here. And over to France. Over to France. Back home. Away, back home. Away, back home. Okay? So you can see there's a pattern. I go out and I come back and I go out and I come back and I go out and come back. And if you had asked me also when I was growing up, if I would be back living 22 miles from where I grew up, I would have said absolutely not. So there are often surprises in the life, and actually putting this together made me realize that that circle that I've made kind of out to different places and back again. So I'm going to tell you a little bit about that journey. Okay, so some of the moral of this is you can go many places and you can always go home. So these are all pictures of places in Michigan. And in making this journey lecture, it really made me reflect. So my favorite season is winter. And it's winter because you can see the patterns in things. And so in particular, the patterns that are hidden in the exuberance of the rest of the year. And I feel like a journey lecture is kind of a way also to do that, to stop and look beyond your normal running. So I was born during the Cuban Missile Crisis. So you can see all of these missiles aimed at the US. My father wasn't sure the world was going to be there the next day when he came to the hospital to get me. So you can imagine the fear of having a newborn and not knowing what's going to happen in the world. This is a picture of my father and my mother. And this tells you basically everything that you need to know about it. And it's amazing because people did not take as many pictures then as they do now, right? There was not like hundreds of pictures, and yet both of them were captured. 2.5 years, well, long enough to get the camera out and do it. And what you see before you are two people, so they both grew up near Detroit. My dad, Dearborn, my mother, Wyandotte. They met evidently, perhaps in high school at a dance when somebody came to the other's school, but they didn't meet again until they got to Eastern Michigan University, where they were both, I think, originally studying to be teachers, and then my dad continued on another path. So they are adventurous, they are athletic, particularly my mother. She pitched into her 60s on softball team. And Here's a classic picture of them now. So my mother is sorting and my dad is looking. This is how, there were amazing amount of pictures that I had where my mother is working on something and my dad is just looking around because that's what he does. He loves to look. So my mom was a teacher for a while before we were born, and then she went back to school. And it's amazing because Mike Elliott's mother sounds a lot like mine. Mike was telling the story. I'm like, wait, that's my mom. So she was a librarian. She loves to catalog things. She's an archivist. She's a musician, a very good musician, does singing, plays multiple instruments. And this is a library, and it's by the dint of my mother that we have that library built in our town. Okay. The picture on the other side is of buildings in Detroit that were constructed by work world who grew up in Clinton. My mother did a traveling exhibit that went around Michigan. My dad built like the pieces to it, and my mother arranged it. It was funded by the state of Michigan. So she is a finisher. It is the one genetic trait that I wish I had inherited from her, that I did not. My dad ended up being a history major, but he was a self and family taught engineer. That's how he made his living throughout his life. He loved looking at things and art, and he is also a musician. So my parents, at 85 years of age, played together in multiple bands. My mother runs the archive for the Clinton Historical Society, directs many people, only now walks at a pace that I came up with, and she's shorter than I am. My father cuts down trees, jumps off 17-foot waterfalls, never met the public, major house repairs. They are incredibly active people. Okay, my dad got pneumonia after doing that. But I went to a seminar on retirement, just out of curiosity this week. And when I'm thinking about, they said, probably your parents, will not be a model for how you would want to retire. I'm like, they are exactly a model for how I want to retire. They are incredibly busy. That is in fact why they are not here today. They both had other commitments through bands that they had made long before and other things. So they are here in spirit with me. So my paternal grandfather was a self-taught engineer. He stopped school in 8th grade to provide for his family. And he built tunnel machines. So you can see here one of the machines that he built. It's all through his field. So they tell the story that he had to build a machine that somebody else designed. And so he built the machine, right? They gave him the plans, he built the machine, and then he made an X on the machine. And he said, this is where the machine is going to break. And he made the part that would help it to keep going when it broke. So you put these tunnel machines down in the tunnel, once they're down there, you're out of luck, right? It's a huge thing. You stop it. Today it would be millions of dollars. The machine goes in the hole, it starts, it breaks. My grandfather brings that part that he made to fix it, and it runs. So he had a feeling of predictive ability of things that got passed down throughout different people in different ways in the family. My grandmother cut books for the company that he had, at least for a while, so she had a head for numbers. This is a picture of me on the bottom there, my mother, my mother's mom, and my mother's father's mom. So my grandmother she worked in shops. She was incredibly able, as is my mom, to extrapolate from a sample size of 1. I grew up knowing there had to be something wrong with that, but just couldn't figure out what that was. My great grandmother, I only knew for a while I was young. And the only thing I remember strangely about her is that she had a double chin, and she told me pointedly that I needed to do this like every day. That I avoided. Now that I'm getting a little double chin, I'm like, I should have listened to her. But there was a lot of love. We would spend summers at my grandmother's, and her husband's, my step grandpa at the lake. And so there was a lot of joy on both sides. This is my sister Sheila and my brother Art. My sister is a counselor in the middle school at Kansas. She can do whole schedules of people, like hundreds of kids without a program. My brother is an engineer at GM, so he predicts when cars are going to feel. There is a predictive gene in the family. So this is in fact my family on my dad's side, and you can see me with the arrow. So everybody in blue is an engineer. We are packed with engineers, even some marrying into the family, right? Are engineers. And these people in red are all the people involved in academics in one way or another. It's actually quite striking. My aunt may have been, I just didn't get a chance to check. So from homeschooling, K-12, teachers, counselors, to university professors, we are just packed with engineers and teachers. So this is a true luxury, right? Because it meant the further I went in my schooling, I stayed very much in the norm of my family. That is not true for everybody. When you come out of a family that does not do academics, you can get further and further in some ways away from that norm. So this was a true luxury, which I think I've only realized more as I've gotten older. I also get to work and raise my son where my family is located. And that is also a luxury for someone in academics to be able to be at home and have an academic career. My family tree, okay, I do genetics, also has a lot of other stuff in it, like everybody else does. You can imagine, given the engineers, there's autism, sensory processing disorders, other sorts of things, as all families have, and they deal with. My mother takes care of my step uncle, and I will take that on when she does not. So families bring also responsibilities with them. So in my childhood, I loved to read. I mean, I was found with a book whenever I could. I loved to be outdoors. This is the first house that we lived in. In the genetic lottery, however, I was not given coordination. So I was picked last in every single sport I ever played. A second to last. Occasionally, was somebody worse than me. One time in my life, I was picked second, and I still don't understand why. You made a mistake, just sure, take it back. But I love to do sports. I also grew up in a house with paintings, and by that I mean lots of paintings. And so when I go to a museum and I look at art, I have a really different view from other people. And the only question I ask myself, which is really narrow-minded, is could I live with that painting in my house? I don't care what the time it was made. I don't care about the history. I don't care about anything. I just want to like it enough to be able to live with it. And I think that it's that looking, which makes me like plots and grass so much. It's like, could I live with this grass? Does it say everything? They're smiling here. Does it say everything that it could? And it comes from my childhood of looking. My dad and I would play game, actually, where we would take this little thing that we call the zipper. And so we would sit down in a room, and then you would look around the room and you decide which painting you want to take home. You go, and then that was the one that you were going to take home with you. Schooling. So I went through school with the same 90 people, give or take, for 13 years. So this could be either a really good thing or a really bad thing. And this is my middle school here. It was a pretty good thing for elementary school. Good teachers, good going through. Middle school, not so good. I was a pretty geeky kid. By high school, this was the 70s. This was drugs and rock and roll all the way. And the closest I came to that was really in school musical, in a very structured environment. So I really liked biology, geometry, gym, I took that every year, independent reading and choir. And I was in I really loved being in the musical because it was a place where I could be with others and sing and dance. And I think that's in part what makes me comfortable being in front of people now, is having gone through that. Okay, so I went to a summer program at Michigan State. I think it must have been like between my 10th, I think it must have been between my 11th and 12th year. And I found my people. There was a girl there who wanted to be an epidemiologist. Like there was another one who wanted to do research. I was like, these are my people. It took me another 10 years to find them, I guess. Okay, so the careers I contemplated on graduating from high school: doctor, teacher, interior decorator. And you may be saying to yourself, interior decorator. Hey, out of all the things that I do now, the interior decorator is not the least, right? I really like those crafts. I do things related to medicine, and I love things specifically, mentoring graduate students. So I went to Albion College. I started out in biology. I didn't really like the teachers. I liked the chemistry teachers better. I did that. I did not fit in with the preppy student body, and I should have left. I didn't. And I had no love of math. I got my math done and I was done. So unlike every other person who has spoken in front of you, I was not an advocate of it. So one good thing that happened out of that was that I was unconnected enough to the campus that I wanted to go abroad my senior year. And so I went to Grenoble. I couldn't speak or write French when I arrived. I had 2 years of French, which meant basically nothing. People went to ski and become French teachers. I went because I wanted to go somewhere. And one of the comments that sticks with me is when they were trying to teach me, because everybody else could actually write pretty well, was how can you speak so well? Because I could pick up the language and write so poorly. I went to the first semester, but I actually stayed a year and I worked with no hair there. So this is a list of all the jobs that I had been to by the time I got to the point of going to graduate school. So you see a lot of babysitting, a lot of kind of manual work, many different things. And I think that this is also something that has changed now with kids being more career oriented, that ability just to be out there working. And so two of these stick out for me. So one was agricultural level tomato planter. This was by far and away the hardest job I have ever done. My respect for people who do that type of work is enormous. The other one was working in my father's shop making tunnel ribs and other things. So I know how to use blowtorch, brick punch, bandsaw, 10-ton overhead crane, fork truck, and operate a press that makes it a horrendous amount of noise to make tunnel ribs. You start with an I-beam up there on the top, and it turns out looking something like those ribs that you can see holding up that. I also cut all the parts for the bed of a railway car. When I walk by buildings that are being made, I love the smell of the welding. It brings back a very fond memory. It also was great for many years in the department, like you would say, who's this person based on their job? If I used any of those, nobody ever asked me. Now you will, but okay, so cell biology years. So I wanted to get a PhD to be able to teach, not to do research. I wanted to work on mitochondria. I wanted to go somewhere that was not a small city, was white middle class, that could quite succeed in the places I applied to make that true. I applied to these places. University of Boulder rejected me. University of Wisconsin didn't get one of my references. I was accepted to Brown and Johns Hopkins University School of Medicine. I decided to go to Hopkins, and I want you to see what happened the first year that I was there. Right? If I had been a student in this department as a master's student, there is no way you would have taken me for a PhD. You can see in the red, I've highlighted because it's really dark. This is my transcript. Those were 2 C minuses that I got. So if you got a C, you had to take the class over. One of the classes was bioorganic mechanisms. I walked in, the guy who taught it was a very tall Englishman with a white scalloped mustache. Full kind of beard type of thing. And I walked in and he did this to get my grade. And he was very good. It was a D minus. I have never been so happy with D minus in my entire life. I did not have to take that class again. Things picked up from there, but it's all to say that grades may not reflect your ability to do the work that you want to do. In my case, there's very little in that class that has anything to do with patterning, anything to do with the stuff that I was wrong about. My PhD advisor, Ann Hubbard, who was a classical cell biologist, cell fractionation, electron microscopy. She loved polarity in epithelial cells, which I love to this day. She was postdoc at Yale with George Pallotti, who had won the Nobel Prize. And she was the fourth tenured faculty member at JHU School of Medicine History out of hundreds, probably thousands of men. Okay. So we were still in that time. So this is what I learned from that experience. There is an optimal angle of similarity with your PhD advisor. Okay. So you start on this side, you're too close, you go over to the other side, 90 degrees, that's way too far. You want to be somewhere where you have some differences, but not too close, not too far. Anne and I, the skill that Anne had in part was critical thinking. So if people were in there, oh yeah, that's really cool. Oh, she would help them to come and think critically. I went in there with critical thinking. And what I needed in part was somebody who said, that's great, keep going, you're doing fine. So we made it through, and it worked out, but it was a little too close to fit that. So hundreds of rats. So one thing that kind of gives me nightmares now is that I killed hundreds of rats during my PhD career. I became allergic to them, so allergic to them. I had to wear a mask. I had to put everything over my skin before I went to get them. We had to keep them in another room at one point. I would take Benadryl if I was around them. Lots of rats in my house. And those hundreds of rats resulted in papers. This paper in particular, this is my first paper. So it's looking at proteins that go to the plasma membrane and seeing how fast they degrade. To do this, you actually have to inject the rats with radioactivity. So you're dealing with rats, radioactive rats. I don't think they'd let you do it now, because nobody's real careful about that, but that's what it was. But I liked it because it got me thinking about rates, right? So this department I was in at that point was a fantastic environment. I have never, and even though this is a great department, I have never been in a place that was that good again. And there were several things that made it that. It was very small. So there were 7 to 11 faculty members, many women faculty members, right? Cell biology attracted women because they liked the looking, I don't know what it is, but it attracts women. Everybody went to every seminar, and there were seminars almost every day. Outside speakers, I mean, the world leaders every week, department journal club, every student, every year, seminars from every student, every year, faculty, students. Saturday, there's some downside stuff. Saturday looked like a weekday. People were there as much on Saturday and often on Sunday. And there was an incredibly intense self and surrounding pressure to work all the time to do that. So friendship. One of the, if not the most important things about graduate school is friendship. We just had a blast. We did ballroom dances. We danced at the annual meetings. We had a golf club team. We made a lifelong friendship. So here are 2 friends of mine, Gudrun Dirka in the top, and Carol Taft, who I'm still friends with today. So it's a wonderful time. So my first postdoc, I ended up in France. Actually, I was going to go to a postdoc in worms in development. I had it all lined up. And then somebody who worked in our lab said, I have this position for a year. Would you like to come? And I said, sure, I already speak French. And it would be fun to go away. In fact, when I did my PhD, I thought, this will be cool because I'll get to do a postdoc in Europe. That was one of my motivations for doing it. So I fulfilled that. I worked with Michelle Maurice. I couldn't find a picture of her, interestingly, and I lived somewhere I had to walk over Montmartre to get to her. So it was a wonderful thing, and people rarely, if ever, worked on the weekend at this place. So my first 4 weeks there, I would go in diligently on the weekend. Sometimes the lights were off. It's like the security guy was sitting there. There were no, lights almost off and nobody around. I finally said, nobody is here. I am not going to come in on the weekend. And in some ways, it was a very good counterpoint to the place that I had been before. There also was no scientific life there, and I was kind of dulled after my PhD, but one day I woke up, like when I recovered from my PhD, I was like, there are no seminars. There's no club. There's really no anything here. So it's like going from one extreme to the other. The person I worked with was very good, but the surrounding environment was the polar opposite of what I had had at Hopkins. So it was useful to see. So one day I woke up in Paris and I said, I can do this work. I have cloned a gene. I don't want to work in the lab anymore. Now, this was no real big surprise because I already had those doubts in graduate school. And everybody who knew me said, you just have to go on and do a postdoc. Don't give up. Just go do something else. It'll work itself out. And one day it became very clear to me that was not true. Now, I had a roommate in graduate school who went there to do epidemiology. So I started reading her books that she left around and thought, it's interesting. So I'm now going to tell you about the epidemiology here. So I applied to Michigan and got in after convincing them I didn't have to take the GREs again. My mother stood in line with documentation to convince you of them that I was still a Michigan resident if I had been traveling around for a very long time. We did that. I lived at home. I did a 1-year MPH, and I landed on the year where all, and I mean all of the historically great teachers were retired or not teaching the year that I was in. So it was a lackluster year and very different. I know I was told that it was very different from the usual years that were there, but that I just happened to land that year here. It did though get me started in epidemiology in a very good way. So I did my master's thesis with Bethany Foxman, and she said, you should talk to my husband, Mike Beatty, who works in type 2 diabetes and genetics. And I said, thanks, that's not the direction I want to go. All right, this story will continue, obviously. So I decided I was going to take a job in epidemiology as a staff position at MSU. And it was this program that was trying to get people to think about cancer and diabetes together. So one of the pieces I had to do was to talk to many faculty members, which was me, who did not like calling people on the phone. I could phone call and convince people they wanted to talk to me. And that was a great education in being able to talk to people. I ended up working also on hypertension, Zimbabwe data set, and being a project manager for a breast cancer study in Polish women with Dorothy Papa, who became a lifelong friend. And this paper, so this was like in 95, 96, 98, that I did this work. And the paper was published 2021. This is a very long gestation. So if you see it on my CV, you're like, where did that come from? Well, it came from 25 years ago when it finally got published. And it was looking at the difference. It was a really cool study design, so small. But Polish women who stayed in Poland, Polish women who came to the US, and how their diet changed in their consumption of sauerkraut. And finding a relationship between the consumption of sauerkraut and the risk of breast cancer. So whenever one of my cases is taking a long time, I say to myself, it's not going to take 25 years. So Dorothy was Polish, and she knew Andrzej Polewski, who was at the Joslin Diabetes Center at Harvard Medical School. So I had become interested in truly working on diabetes at that point, and he worked on epidemiology, genetics, and type 1 diabetes kidney disease, which sounded great, but I did not want to do genetics. And so when I went for my interview, I said, I really want to come here and do this, but I do not want to do genetics and PhD here. And partly it was because you had to go back into the lab, and I knew I didn't want to go back. So I did a lot of learning. I took a lot of classes at the Harvard School of Public Health. I got the chance to work with Louise Ryan on a paper there. And I took actually the generalized additive model, which I used in the paper. So that was a lot. So I also did my first statistical genetics paper after saying I didn't want to get work on genetics. I got pulled in with the TDT, or the transmission distortion test, because we had a lot of people. You always like so transmission distortion is two parents and then an affected kid. But when you have diabetes, your chance of getting kidney disease is exorbitant. So it's actually the people who don't get kidney disease that are unusual. And so it was kind of flipping that on its head. And the nicest thing about this paper was that Richard Gilman, who was a well-known geneticist, he actually wrote something on it and said, basically in review, clever idea. That's a clever idea. And he signed his name. So that let me know and gave me an enormous amount of confidence that perhaps I could actually do this. And around that time, Steve Rich, who I don't have a picture of here, came and he said, I started looking about postdocs, and he said, you should think about doing a postdoc with my Becky. I had a couple other people who I've been talking with Louise. Louise Weinberg came here to interview finally with Mike, and moved into statistical genetics. So when I came here to interview, I really liked the atmosphere of the students. I was so incredibly impressed with my interactions with them. I became part of the Fusion project, which I still am today. So extra thing in there, from the United States Investigation and NIDDM. And then we got to the point where, you remember when I told you about the PhD advisor and that angle of similarity. So Mike is, or was much more at that point, a tables person. I am a completely pictures person. And so we worked to come to a language that was somewhere in between those. And I think he moved more toward pictures than I moved toward tables. So I do say that they're useful. And so that became a very long collaboration together. And it brought me in contact with many people who have been just wonderful to work with. So I want to start off with staff, because these are the people that really do the brunt of the work. So Heather Stringham was here, and Jackson joined the year after I think I arrived. So these are the people that keep the work going from year to year as graduate students come and hopefully go. You have to have some way of keeping continuity, and in large projects, it's just been a pleasure to work with them. There have been many postdoctoral fellows, many students. I don't know if I got all the names up there. I've worked with students between 1 year just for their master's, and some fortunate enough to work with for their PhD. So this has been really an amazingly great part of being here. That initial impression of the institute students never left. So the first thing I did was linkage analysis, okay? An almost pointless character building exercise. And that is because we were looking under the lamppost. So at that point, this is, you look at a family, you look and see what people are sharing for their genetics. So it was a commonly held but wrong assumption. So we were not the only ones doing this, right? And there was nothing particular about Mike or about the direction. This is all you could do at the point. And there were questions about how many causal variants are there? 1, 2, maybe 10? Okay, we just didn't know. And if you want to do power calculations, you want that number to be like 1, 2, maybe 10. But there just aren't rare variants with strong enough effects to produce linkage peaks. So almost nothing ever came out of all that work. But it did build the type 2 diabetes consortium, and this is going to become important. Next, candidate genes, another character building exercise. Okay, so many small studies of type 2 diabetes, 100 to 400 people. We had one that was somewhat bigger, testing 1 to 10 variants and hundreds, maybe hundreds of genes, one by one. People's best guesses from biology. Biology is not a good way to make a guess. about the gene that's going to be important genetically. We didn't know that at the time. There were hundreds of publications published, hundreds of genes found based on p-values less than 0.05. 4 or maybe 5 out of all these genes actually were truly associated with diabetes once you got study samples big enough to look at them. So again, it was a learning experience, and it was good because people collected type 2 diabetes case control samples, almost all European ancestry at that point. And many genetic epidemiologists and statistical geneticists were trained. Okay, so there can be good things that come out of things that don't actually yield scientific value itself. So around this time, actually probably before this time, Jack Helflash, when he was chair, he said to me something like, and I'm taking this out, but I hear you asking good questions in seminars, would you like to be a research investigator? Okay, I said, yes. So many, many thanks to Jack for that. I also heard from Morton Brown, who had a very particular idea of what a biostatistician looked like. And he asked me, as I happened to be walking home next to him one day, when are you going to look for a permanent job? And I said, I like it here. So your value to anywhere that you go, right, especially if you cross boundaries, will depend on what people see of value. And you want to get yourself somewhere, hopefully, where you bring value that's not there, and where your value is appreciated, where you go. It's kind of it's a little bit of a tricky balance there. So ways to make a big splash in statistical genetics. There are 3 of them. Really new technology, can be on a small data set, really big data set, can be an old technology, clever idea, preferably new technology data. Okay? So the way to get a science paper, you do all 3 of those. Okay? So we had a new technology in a wide association study. We had, when we put all these studies together, 3 different studies together, we had the biggest sample size. And we had the advantage that Gonzalo had been working on imputation. And so we could test many variants, and Kristen Willer, who was a postdoc, his name I realized, but didn't get up there to apply that to the data that we had, so extended it. So this was a set of 3 papers in science. It was one of the most exciting statistical times of my life, because when I looked at the genes, the variants that were associated, one of them was the colleague Karen Mulkey. So I called her up at night. I said, Karen, there is a coding variant in the leak transporter in the islet beta cells. Those are the cells that make insulin. I'm like, this is a real new variant that we have found for the very first time. Turned out somebody published before us. It was really exciting, you know, for that moment, and just that thrill, truly, which I've almost not replicated in my career, of saying I found something that nobody else knows. Well, I did, okay, but that I think nobody else knows, and that team has gone on to deal with just multiple insights. It also, so Ryan Walsh is in the audience. So it also yielded the plot up here on the top, which spawned 1,000 plots. That is more than 1,000 plots. So this was the picture that was the precursor of the locus zoom program, which Landy Time started on. Ryan Welsh has now taken forward. Andy Button has worked on, and this turned out to be the industry standard going forward for everybody. The word for it is locus zoom. So that was really cool. So I get bored. RNA sequencing was coming into being. And so I went on sabbatical to the University of Chicago with Jonathan Pritchard. And it was part of my transition to RNA expression and other molecular trait work. It was a really fun lab group. So kind of getting me into colleagues, you know, who were doing that kind of work. On my way over to Chicago, I finally decided that I was going to adopt. So there is something that is very nice of getting yourself out of the place where you've been when you're trying to make a hard decision. So this had been a very, very long journey. I had been contemplating this for many, many, many years, and had gone through thinking about many different things. And finally, it fell into place. And in part, it fell into place because I realized that when female friends were having babies, that I didn't want to be around. And that thought that I could not support people because it was so strong in me, got me to say, you need to go ahead. And so, this is my son. This is the day that he became legally mine in Ethiopia. You can see I'm dressed all in white. I'm gonna embarrass him. He's sitting right here. He's growing a lot, as you can see. And that was one of the most joyful moments of my life, I would have to say. These are some pictures. This is the first exposure to snow here, because you hadn't seen snow before. This is with my father down there. This is with a friend of mine, Deanna, actually, Marriott's daughter, Ivy, going up north. So lots and lots of fun going through life together. These are the cousins around some birthdays and Thanksgiving, so we're always all together. And he is a very athletic and gifted kid. So there have been hundreds of baseball games. And I have to say, I am so grateful that it is baseball and not, it's outside, it's slow, you get to talk to people, it's not inside where it's loud and balls are bouncing, and it's absolutely the perfect sport. I think he would have preferred football, but I wasn't willing to let him do that. Okay, so the last shift has been into a more statistical cell biology and into DEI. So this is a picture. I've come back around to cells. So these are nuclei from cells. So you can see here on the side with all the kind of globs of color. These are different. Different cells, or nuclei from cells that are being clustered based on their gene expression. And each color is a different cell type. And you can see in the picture on the far side from me, you can see the proportions of different cell types that males and females have. And this is work from a paper that Sarah Hanks is doing, and it's based on data originally from Rishi Varshni and Steve Parker, who generated the data. And so this has brought me all the way back around to cells and trying to understand what is going on at a cellular level in response to phenotypes, in response to proportions of cells. I would never have imagined when I started this that I would come back around this far, but I have. So diversity, equity, and inclusion work. So this is kind of my takeaway of doing this. So you have to jump in, but then it takes a very long time to learn how to do this. It is a continual learning, and it is only, I think, work that you can do really if you are learning and reading and listening to others is just crucial in doing it. If you do it without doing those things, life can be hard for the people who are on the receiving end of that work. There are two other things. So you need to have lots of support. If you're going to do this work, you need to have a very strong network of people who are doing it. You need to be taking care, as you do with everything, of yourself. And you need practice in different avenues, not just one, because things kind of bore you and give you opportunities to learn. So I'm part of up here, the Aspire group, which then our parents intent on racial equity, which is a parent group that works to make Ann Arbor schools better. I'm also part of Quaker Meeting, where I'm on the personnel committee, and we're writing actually a sexual harassment, or we'll be writing a sexual harassment policy for the meeting. So it's been very interesting to think about kind of what the university has in place and what you can do if you actually can start from scratch and decide what you want to do. And the advantage and disadvantage, quite the meeting is done on a sense of the meeting. So there is no top-down anything that can happen in that context. I also take Tai Chi from Orthodox Young at the Peaceful Dragon. I listen to the Centering Justice program, which I would strongly recommend to people who want to get a sense of being able to listen to people. My takeaway. So it's the people that make the journey worthwhile. That is really in the end what you have. And you may have no idea where you're going, but you will get there if you listen to yourself. And each time I changed, there was something inside of me. It was very quick, actually, almost every time. There was something inside of me that said, I want to do something different. Finally, I've been thinking about it for a while. Go, and often within like 3 or 4 months, I was doing something different. That's easier to do when you're younger. So if you do take this long winding road, you will find you have delayed money making because you keep being a student for a long time. And if you come out with large debt, that probably is part. I was lucky in that college was cheap enough. My parents paid half. I had a scholarship for part of the most of the other half. So I came out of school without debt out of undergrad. If I had not, it would have been a very different story. And having a broad view of an entire field may actually bring you back to where you started. So I've come back to South, where I started. And that is completely, I think, unexpected, but quite something. Thank you.
Thank you so much, Laura, for that inspirational journey. Any questions for Laura?
You have a picture of yourself doing a headstand?
My parents had a 50th wedding, and 40th, 50th, I don't know, wedding anniversary, and we all tried. I'm not sure I was able to stay up long enough to get a picture. You have to know that my niece was a college level gymnast, right? The genes went on, but not quite to me. Many of my family. Yeah.
Of the something like 16 museums on campus, which one do you recommend?
I like actually, so I haven't been to all of them. I like the archaeological museum, the Kelsey Museum. So I like museums that are small. I really love pottery. They've got pottery there. So that's probably my favorite. Yeah.
I feel like you're on your 7 to 10 sabbatical clock. You might be coming up in the middle or towards the end of it. Do you have an idea on what your next step is?
You are very perceptive. It's really interesting because I told you I'd gone to that retirement seminar just out of curiosity. It's really kind of what I'm going to do for the next, because it's probably 7 to 10 years. I'm 60 right now, that I'm going to work. And I'm not sure. I mean, I've done the DEI work in the department, and I like that work, but it takes an enormous amount of effort. Right? It really, I'm not sure that's something I could do full time. I'm not sure kind of what the place would be. I really think I like policy and like having things be clear. So maybe there would be something there. I'm enjoying being in the cellular world again, and it's all of those changes within statistical genetics are not quite far enough apart from each other. So I think I'm just waiting to see whether there's another 3-month period where I go, that's what I need to do, or whether we're gonna kind of push that boundary. But I feel it, and I feel it Yeah.
What art do you have in your home, and have you ever framed one of your favorite drafts?
That's a great question. So mostly, I have art that I've done, in my home. So I do, I've taken classes in pastels, in watercolors. I did art in college because of PCAM. I, I am, I am, I have really strong opinions about the art, and so there's actually fairly little that I really want to have in my house. So I had a local artist, a print of hers. I have a picture that I got from my parents, that hang in the kitchen, but perhaps surprising little beyond my own art in my house. Do you have another part to your question?
Oh, yeah, like, do you ever end up framing one of your favorite drafts?
I have packed them up, yeah. I really think that some of the things that people have made are quite beautiful, and when you make a beautiful graph, it makes such a difference because you can communicate so much information. I could say I might go overboard, probably every student I have might feel that way, but it really makes a difference if you can do that effectively. And in fact, I had one of my most ironic moments was I was talking to Orly McCall, who's was an editor, I don't know if she still is, at like Nature Genetics, and it was on one of the papers that I'd done with Mike Gonzalo and Andrew Skoll, and she said, you know, that paper that you published had such wonderful graphs. It must be so wonderful to work with Gonzalo, you know, and I'm like, that's not Gonzalo's graph. In that paper. So the graphs do get attention, and they do work. Yeah, Kathy.
Hey, Laura, really enjoyed your talk. I think that one of the cool things about the journey lecture is kind of like what we learn about ourselves in the past. So it's really cool to see that. But my question is, and I think particularly is directed to a student issue, is that when you move into an area where you are not a perceived expert, so that, you know, we talk a lot about valuing multidisciplinary research, but maybe not when it's in a department, right? So what would you say to students who want to follow that path? How did you kind of keep it together to say, like, well, I'm going to stay in biostatistics even though I don't have a stat degree?
Right. I think there's two pieces of that, at least. So one is realizing to yourself that being in that environment is useful. I knew that it was useful to be there, to hear the things. And I can feel like when results are wrong, I'm a really good, I knew that I was a really good applied statistician. And that I could actually look at other people's work, methods work, and also see when it was wrong, even though I couldn't understand the equation. So I knew that about myself. The other thing is, I mean, there certainly were people who did not think, I think, you know, that I had the right skill set to be here. And it really is understanding the place that you go. And I think an enlightened place will make it clear, right, that we are looking, this is valuable. If you get yourself into a place that isn't going to go there, then it's just going to be heartbreak, right, for you. And I think our department also had that, like on the computers, when people went more computer science, right, there was also, I think, kind of this angst, you know, that it was getting away from the pure methodology. I mean, it turns out to be part of what saves us, because we get the health data science program, you know, out of it, we get all the statistical geneticists, you know, that do that. And it's really how forward thinking is that place that you go into, right? That they say some departments, I see them, right? They're like, we want diverse people. Like you have to, I don't know, if I had asked when I first came here, you know, I didn't ever think of, when I first came, I wasn't thinking of staying, right? That just kind of happened along the way. But if I had, I'm pretty sure the answer would have been, you don't fit our criteria. You know, it's nice you're getting training here, but you know, you will not become a faculty member here, at least for some people. So it's a hard thing to know what to do there. I do think though that having that ability to go across disciplines, like I can read all the way from the biology, I can read up to the epidemiology, I can do the study design, right? That's very valuable to think about the epidemiology. I know how to understand the statistical genetics. So whereas some people get stopped, I can go all the way across that. And that is just a wonderful thing. And at some point, you just have to believe that this is what I want to do. And if you don't fit enough, you just have to leave. Right? You just have to pack up your bags and say, I will go somewhere where people value what I do. Yeah, and you
Just out of curiosity, was sauerkraut consumption?
yes, it was. Yeah, all that 25 years, and the answer was yes. There's a very nice dose response to consumption of sauerkraut and decreasing risk of breast cancer. Thank you for asking.
Other questions? All right, Sarah, you need to present the certificate to Laura. The most, in order to acknowledge her contribution and sharing not just today, but many years of her journey with us. And Laura has certainly taught me, and this is something that I think is true for everyone, that start where you are, use what you have, and do whatever you can in any point when you want to take a challenge. So that's how you get started. And as a department, we have a long way to go, but we are a better department because Professor Scott is a faculty member here. For that, she deserves to get drunk. And this is the feedback for Laura and you to enjoy. Thank you very much, Laura. To all of you, have a wonderful afternoon. Keep the spirit alive as you transition into the weekend.
And thank you to all of you for having me in this department.
Just one more thing which makes this day special is that to all of you who participated in the Biostat Carnival tournament, we had a crushing victory. So thanks for everyone. Have a good weekend.
Yeah.
And what you like?
I'm gonna be playing. I would be playing. Or maybe I'd be. Yeah, maybe. Yeah.
I love Get on something to do with yeah.
So are you still pitching? I have to turn. Right, it also clearly got better. I know. You're starting to take a game.
This summer is gonna be really hard.
Your son?
Yeah, like my son is 17, and he's a year in school. He actually just signed for their baseball coach. And
You say stop that, like students, then they would stop that.
all of them.
And I just feel that they want
all of the
all like every.
Well, yeah. So is that? You know what I mean? Yeah. It's just been too much. So that's great. We will do everything we can do. I think it's fine.
Yeah.
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 Yeah, he's a return short.
Yeah.
He plays third.
Yeah, that intro. We'll get excited.
Oh, nice.
But, and so.
Because you can't do this.
Who knows? So, when they talk. But you have the teacher, so they'll start at the junior.
Yeah.
And then she said that as you pitch, you'll probably pitch for another class.
Yeah.
And they do it. I said, well, that's great. I'll give you one of these.
Yeah, they work on all those.
Absolutely.
But
I did the class, all you saw is my partner. And we're playing to see. But I don't know if this.
Yeah. And it's, yeah, there's also like, MSA. Yeah. It's nice.
Your favorite team.
Nice.
I really, I like to take a look, because maybe.
Yeah.
If you're on the level.
do a fair amount when there's a lot of.
We're playing.
Yeah, I stayed away from almost all.
I don't know, but 18 is the much, the pitching is, the pitching just gets on two different levels. You know,
Yeah.
it's-
Oh,
Yeah. So,
he's at 83. Right.
okay.
I was tempted to look at my phone.
He's sitting at 83 and he has a fast foot.
Yeah.
It's like,
Yes.
and he's
Yeah.
working out and doing everything
Yeah.
he can do. But
So
I
much
said,
that,
you also have it in the front, but having like,
Yeah.
it's a little bit different.
Yeah.
too, and it's good.
you know, on that, but that piece. was-
You know,
We're looking similar to that.
Yeah,
You're right.
I need to
Yeah.
seem to
Yeah.
serve. I'm not.
We're so central to.
Well, thank you for everything, and I will be definitely.
Yes.
How's it going? How's it going? Especially
Yeah.
if I love.
I remember this person, environment, you know, way off.
Yeah.
Yeah, I don't, I kept going,
Yeah.
Oh, well, good luck. Maybe we'll see you this summer. It was nice to meet you.
Well, there's so many people on that 2007 paper that put up there that I knew were.
Yeah.
I didn't know that they were all on that paper, but
Yeah, and I didn't actually say.
even Peggy White was